And hey, microbiology fans, Dr. G here with just a few bits and pieces of things to pick up on from the end of Chapter 4, talking about our basic cell biology. We only have like 3 or 4 slides to cover today. And next week we're going to be getting into some ways of looking at different bacteria and how they use different material for metabolism. Talking a little bit about metabolism. And we'll start thinking about the first quiz, exam, whatever you want to call it. And it's going to be asynchronous, but it's going to be a Moodle thing where you have an hour, and it's going to be pretty easy. I'll give you some good review notes and things as I start seeing what we're doing. Let me know if you'd like to have some kind of zoom-in review. I don't know. Main thing is that it's going to be open book, open note, open everything, but you only have an hour to do it. And so if you're not reasonably familiar with the material, you may not have time to look everything up online. So what you need to do is go through your notes and think about the topics and points we've hit. And as I say, I'll put together at the beginning of next week an outline of things I think we've covered that are critical. And we'll see where we go from there. Okay? We'll also have, it's been sort of a tradition of one of the extra point things is that you're going to find an article, a review, that you can review, write a 1 paragraph or so review, half page, of an article pertaining to the area of microbiology that we're studying, which is basically cell biology and basics of the discovery of bacteria. So we'll see where we go with that. Anyway, so when we finished up, we were talking about things in the cytoplasm, specifically the ideas of the ribosomes. Ribosomes are critical machinery for protein translation. They are required to take mRNA and translate it into protein. And one of the really cool things is that both you and I, eukaryotes, we've got ribosomes and bacteria have ribosomes. We both translate RNA pretty much the exact same way. There are some interesting differences though. First off, if we look, and you don't need to really memorize these numbers, but just recognize that ribosomes always have a large subunit and a small subunit. I kind of think of it as the hamburger bun model, where you got the top part of the hamburger bun and the bottom part of the hamburger bun, and the RNA is going to end up being I don't know, the burger patty, the lettuce, whatever, mayonnaise, and it's going to be in the middle. And so the RNA is being read by the ribosomes. And the large subunit has got an RNA molecule, and it's about 2,900 bases. Big deal. Anyway, the small subunit has a smaller RNA molecule. And then in addition to having these RNAs, they also have a lot of proteins. This is called the structures are referred to as ribonuclear proteins. And they're pretty big. I mean, you can actually centrifuge ribosomes down. They're so big. So the key difference between eukaryotes and prokaryotes, that is you and I versus the bacteria, is the presence of more proteins and larger RNAs, and in fact there's 2 different ones here, not that it's all that critical to worry about, but the prokaryotes and eukaryotes do have a lot in common, but the eukaryotic ribosomes are bigger and probably just a bit more complicated. Now, one of the cool things about the ribosomes is they serve as a target for a variety of antibiotics, probably some that you've heard of. As we've been talking about this, the cell wall synthesis is a target for antibiotics. Our various illin types like penicillin and ampicillin, amoxicillin, these are targeting the production of the cell wall, of the peptidoglycan cell wall. But then, because of the difference in ribosomes, there are some antibiotics that are going to completely inactivate the ribosomes and block their ability to do their job, block protein translation, which is going to be killing the bacteria. It'll absolutely stop them in their tracks. And our ribosomes are structured a little bit differently so they don't affect us. which is the whole key behind an antibiotic. I mean, it's easy to kill bacteria, but finding a technique or a drug or a system that'll kill the bacteria and not you, now that's a trick. Like heating bacteria up kills them, but it would also do a pretty good job on you as well. On the other hand, we got a variety of antibiotics. The aminoglycosides, you don't need to memorize these structures or these names, but just know that these are the antibiotics can target ribosomes. Streptomycin, kanamycin are two that you probably have heard of. The tetracycline and doxycycline, the two I know of, they're targeting the bacterial ribosomes. Spectinomycin is another antibiotic. So one of the interesting things is that antibiotics that target ribosomes, man, that kills bugs dead. That will stop the bacteria in their tracks, and they will be dead. Whereas the antibiotics that are blocking the cell wall synthesis are really almost something that is going to retard bacterial growth, in that once the antibiotic has been used up, the bacteria that are left, they can actually start growing again. With these guys, it'll often kill everybody on the plate. So these are pretty powerful antibiotics. All right, now another component in the cytoplasm that is a big part of cell biology is something called the cytoskeleton. So it's an analogy that's not really very useful to think of a cell being like a jello-filled water balloon filled with enzymes. But really, there's a lot of structure in that cell. And it allows cells to push on one side versus the other, to actually engage in amoeboid movement. And it's also this cytoskeleton that sort of supports the cytoplasm and allows things to actually have position and not just be floating around randomly. And so the cytoskeleton is providing an internal structure and shape. It's also critical for cell division. That is, the ability to cells to actually begin dividing the chromosome and then to actually pinch off into two cells. That's going to be a component of the cytoskeleton as well, to kind of carry out this pinching going on. It also is able to pull apart the DNA. Now, in eukaryotes, in all the various eukaryotic cells that have the nuclear envelope, the chromosomes are individual pieces rather than a circular DNA like it is in bacteria. And the chromosomes, maybe you remember this from, high school biology, A&P on the street, the chromosomes will line up at the midline, and then they'll be pulled apart during the part of the mitosis known as anaphase. And that pulling apart is being carried out by components of the cytoskeleton. So another component that we don't want to forget in the cytoplasm is the genetic material. That is the DNA and the RNA. So in eukaryotes, you and I, we've got our DNA arranged in chromosomes and stored in the nucleus. But in the bacteria, it's no organelles, there's no membrane-bound compartments. And so you have the DNA sort of spread about, is referred to as the nucleoid, which is more or less a spot in the bacteria where the chromosome, where the DNA happens to be. RNA synthesis is also occurring right there because it's being tracked off of the DNA, and protein synthesis is occurring in that spot as well. So the nucleoid ends up being kind of a spot cleared when you're looking at an electron micrograph. It's a bit clear to the cytoplasm because that's where the DNA is, or some of the ground cytoplasm as it's called, and you see the DNA structures here. Well, the kind of the pile of it. Now, in order to actually see the DNA as a double-stranded molecule, you have to do a real high magnification. And this is one of the things that it looks like is that the host genome, the nucleoid itself, is a really complex, looks like a bowl of spaghetti. And then lots of bacteria will also have what are known as plasmids. And these are small extrachromosomal DNA molecules. And they are carrying little bits of gene sequences, little pieces of DNA that are gene sequences that can confer things like antibiotic resistance. And they're also one of the tools we use in cloning genes, using plasmids. So we can hijack a plasmid and actually take it out of a bacteria, genetically modify it using molecular genetic techniques, put the plasmid back into the bacteria, and thereby add new genes and things. And bacteria do this on their own too. They can swap plasmids between one another. And that's one of the ways that drug resistance and antibiotic resistance is able to move back and forth. Now, a last bit in terms of the systems that cells have. So these bacteria, while they don't organize into tissues, they're not just completely independent, random little dots. They actually can work as a bit of a team, and they also can, as an individual bacteria, begin moving. This notion of what's called chemotaxis. And this is a way of the bacteria can, in a sense, sniff out a food source and begin moving towards it. And so we have positive chemotaxis, which is something where you move towards an attractant. And then there can be things like negative chemotaxis, where it moves away from something that is essentially a repellent. And the way that it typically works is that the bacteria will exhibit sort of a random motion until they get a signal, and then they'll begin actually moving towards it. So we've got this tumble idea where the bacteria are just sort of rolling about. There really is no specific direction. And then there'll be a coordinated movement when they get some sort of chemical signal that'll say, hey, over here, a little bit like a dog picking up on a scent, and off they go. And so when you look at a micrograph tracing the net movement, this is what a random walk looks like, just sort of roaming about in a circle. And this is what chemotaxis will look like when you've got the signal, a chemical attractant, and the bacteria begins to hone in on it until it begins to actually track towards it. So these are some of the ways that bacteria are actually able to interact with their external environment. One last thing. We mentioned the archaea, and I actually was saying, we're not going to talk about them anymore, but just a few points in terms of nerd bits about bacteria. Remember, the archaea are the bacteria that live in real extreme environments, and they really aren't disease-causing. But archaea have a slight different kind of linkage between the polar heads of the phospholipids and the fatty acid tails. And it's a bit of a wonky nerd bit. They have typically you have an ester link. That's what you and I have, and most bacteria, the bacteria, not the archaea, have. The archaea have something known as an ether link. It's just an oxygen link here. It probably makes it a little bit tougher, that they don't fall apart as much. The archaea also have a slightly different structure to their overall peptidoglycan, or their external coating, in that they don't have a peptidoglycan. They've got these multiple kinds of layers of glycoprotein and different kinds of molecules there floating around. And primarily, I suspect this is part of the way they're able to withstand these more extreme environments. Just a little nerd point, but there you go. Okay, so that was the last bit I wanted to cover for this chapter, just to kind of make my way through it. We'll pick back up on Monday on Chapter 5 and start talking about some of the tests we can do to identify bacteria based on the things they eat. And from there, we'll begin to summarize the kinds of things we're going to put in our exam and what we're going to be covering. Give me a holler if you think a Zoom interaction would work. I will put together over the weekend and get to you a list of topics and things that I think we've covered and that I think are important to begin reviewing. And I promise I'm not going to abandon you. We're going to be cool. So that's all I know for here. This is Friday. This is Dr. G. I'm out of here. You guys take it easy.